And get started. So the next event should be, I think, a lot of fun. It's a panel discussion. The way we're going to organize it is everyone is going to give a 2-minute talk. Supposedly, after the end of 2 minutes, I'm going to just advance the slides so that if they're not done, they're done when the next person's slides come up. And then after that, they're going to actually take just a second and move the projector so you can see the folks as they sit here. And then we'll have a little back and forth between the panelists, and then we'll open it up to discussion with the entire audience. Yeah. So the panel topic is, do we need a clinical trial of genomic risk prediction? You know, everyone thinks RCTs are the gold standard for everything, and we're always told we need an RCT, and if we don't, then we don't have good data for any clinical conclusion we want to make. So if the answer is yes, we do need to have clinical trials for genomic risk predictions, then what should it test? Who should it be tested in? And what are the outcomes that we should be looking at? And what do we need before planning such a trial? So we've asked each of the panelists to address this, and we'll see how it goes. So in order, alphabetical, the first presenter will be Muin Curry, followed by Alicia Martin, followed by George Mensa, and then Gina Peloso, and then last but not least, David Valley.
You're doing the clicking. So the clock started ticking down. This won't be as entertaining as the debate yesterday. So in 2 minutes, I just wanted to put the context first, and then we can discuss later. In order to evaluate any genetic test, including polygenic risk scores, we need to answer a number of questions. So 20 years ago, the ACE framework was created by CDC and collaborators: analytic validity, clinical validity, utility, and the LC issues. Notice at the center of the evaluation is the intended use. So you can't have an evaluation of genetic risk scores in general. It has to be for what purpose and in what population. And based on that framework, the EGAP working group, which was mentioned yesterday, which tried to do at least the consumer reports version of what Zach Kohani mentioned yesterday, an evaluation of genetic applications by intended use. The EGAP has come and gone. Some of you are members of EGAP and have taken this work to ClinGen and other enterprises. CDC, on the other hand, was left with using an evidence-based framework to classify genomics into its applications: Tier 1, Tier 2, and Tier 3. Sad to say, genetic risk scores are still a Tier 3 enterprise, meaning their clinical validity and analytic validities are still in dispute. And we can go over that later. Next slide, please. So to answer those questions very broadly, yes, we need clinical trials, but it will have to be multiple trials depending on the intended purpose. And of course, we have to answer all the other questions first. And what we need to know first is within the populations we do the trials are the analytic and clinical validity for that particular application. The type of trials could be test or no testing, or if you've already done the testing, let's say part of Geisinger and others, you can decide to return the result versus not return the results and figure out whether or not genetic risk scores could add value. Or you can do hybrid studies, which will be discussed by George Mensa, meaning a combination of trials and implementation studies. Thank you. 2 minutes? Later. Okay.
Hi, so I want to focus on the question of who. There's a lot of really big, broad questions being posed, and since I have now 1 minute and 53 seconds, I want to focus on the questions of who. And so thinking about the questions of who, I think it's really important to think about where our data is coming from. Of course, genetic data in general is vastly, vastly Eurocentric. About 80% of participants in GWAS are of European ancestry, which vastly contrasts with the global population, where they constitute about 16% of the population. And so to try to understand how polygenic risk scores are going to work in a diverse setting, like here in the US, where European ancestry individuals only constitute, say, maybe something like 60% of the population, it's really important to think about how this would actually translate and how accurate this would be in diverse populations. So this is a plot that you've seen a couple of times now where we evaluated across 17 different quantitative traits. If we ran a GWAS in the UK Biobank and European ancestry individuals and then tried to translate this and see how prediction accuracy performed in different populations, you can see that the reduction in accuracy is pretty massive. It's somewhat trait dependent, but considering we looked across 17 different traits, this is kind of a pretty general phenomenon. So of course, we're doing about half as well in East Asians and about, only about a quarter as well in African ancestry populations. We understand the genetic basis of this. We're building predictors from correlated, not causal effects, and this is determined by things like human history, which influences allele frequency differences and all sorts of factors. You can go ahead to the next slide. And so I think one thing that we really need to invest resources in is trying to couple all of our diverse resources together. So we've evaluated how risk prediction performs when we have genetic data from the Biobank Japan and from the UK Biobank. And in general, what we've seen is that our ancestry matched results are performing best and that there's consistent promise from diversifying efforts. For example, looking in East Asian populations, we're seeing that we're doing a better job. If you look at the difference between these plots on the left and on the right, one thing that we know is that we actually need differences in our cohorts. We need to account for those differences. We need to account for the fact that some cohorts are population-based and healthier than average, whereas others are disease-based, and we need to consider those environmental differences and differences in baseline health. So we need to match our cohorts, and the precision of our phenotypes is really going to matter here. I realize I've gone over time, so.
Great, thank you. I would like to use my 2 minutes to make 2 very simple statements. And the first statement is the title of the first slide, that I agree that we do need clinical trials, and I'd like to emphasize that we really need to address at least 3 types of outcomes. By definition, since it's a clinical trial, we will be looking at patient outcomes. Just going on a portfolio analysis of how our institute has done, we are more likely to support clinical trials that use hard endpoints, not biomarkers, not surrogate endpoints, so taking that into account really helps. The Institute of Medicine also taught us that it's important to look at some of the standard of care issues when we're thinking of clinical trials, in particular equity and patient-centeredness crucial here. But I've put a red box around the implementation outcomes because those are outcomes that we are likely to forget. And it's really important that we emphasize the issues around cost, issues around affordability, issues around acceptability at the provider level, but also at the patient level and health systems level. So those are important. In the next slide, I make the second statement. This is about study designs. I think in this day and age, we really should not focus solely on clinical effectiveness as our goal. We should do a combined approach that at the same time as we design studies for effectiveness, we also collect information on implementation, because many of the barriers and facilitators of implementation we really have to formally assess. Muin Kuru mentioned the hybrid design. This is a classic example. The type 1, where you focus mostly on assessing the effectiveness and you gather information on implementation. But then in type 3, where you focus on implementation, where you have clear, compelling evidence of effectiveness already. We're very, very interested also in type 2, where you're doing rigorous designs to assess effectiveness, but also rigorous designs to assess implementation. Thank you.
All right, so I would argue that we need clinical trials to be able to build up the evidence that this is actually using polygenic risk scores in the clinic is going to have meaningful decisions when coming to making care decisions for clinicians. And to be able to build up that evidence, you need to be able to change guidelines for clinicians. And the way to change those guidelines is through having clinical trials showing the effect of an intervention. So it provides concrete examples of the usefulness of this. Considerations I have for designing a clinical trial is that predictions should be as accurate as possible, so the correct populations to be targeted in terms of both the ancestry as well as including environmental or lifestyle factors when you're looking at the genetics in addition to those factors and controlling for those in the trial. And importantly, this was brought up a lot yesterday, but that there should be an actionable response based on stratifying people into a different risk category based on the polygenic risk score. And just as important as there being an actionable response, that implementation of that actionable response should be feasible. So that's something that we could measure and we can measure accurately. Alicia talked about the ethical issues in terms of doing these kind of trials, but some outcomes, I think both whether to test and when to start screening are good initial outcomes to have for these kind of trials, as well as secondary outcomes on how individuals respond to the information.
Hi, I'm David Valley. I come at this from the background of a pediatrician and a geneticist. Many of the diseases we've talked about are not necessarily in my wheelhouse. I also have as an ulterior motive, I would say, the primary advocate for genetic medicine at Johns Hopkins. So any good data that I can lay my hands on will make my job of advocacy easier. So I think we should think in terms of learning from what's been done before, and as I thought about it, newborn screening for PKU, that was done at a time when we didn't know anything about what was the right, treatment, what were the right ranges. And if you think about it, you tell those parents that their child is going to have a problem, and they look perfectly normal when you make a dramatic change in their lifestyle. What is in progress? We heard about a lot of that in the last 2 days. What are the questions? I think we have lots of different kinds of trials, and we have to be very careful to list the questions we're trying to answer and make sure that the study is designed appropriately. And there are lots of design issues, and most of these have been mentioned by my predecessors. I will make the point for educational add-ons to make sure that the people that are using the trials and the people who are the recipients of the trials have some educational value to them. Next slide. And then what are the outcomes to consider? And obviously we're hoping to get better health. What kind of phenotypic measures? We might follow the reproductive behavior, and can we prevent problems? The economics, I can't stress that enough. If we can't make an economic argument, we won't get it covered in the long run, or this kind of testing covered. And then we have to influence the behavior of our colleagues. We have to convince them that this is a good way to do medicine.
Okay, so I think we're ready to go ahead and move the projector. Folks can take their seats. See, if they had one of those smaller projectors, this would be a lot easier, right? All right, well, thanks very much. So I think those were really great introductory comments. Let's just start by seeing if anybody would like to add anything to what they heard from their colleagues. So, Gina, you want to go first?
I have nothing to add.
Okay. George?
No.
I have nothing to add.
2 more things to add. First,
You're good.
So there are trials and trials. So we heard yesterday about the kind of trial that could be done, which is using genetic risk scores to look at people at high end of risk. And if you're evaluating a treatment and intervention, you can pick up the high-risk groups to evaluate utility because you'll have more outcomes that way. So that's another kind of trial. And I like Peter Kraft's emphasis on the combination of modeling and using the SysNet approach, for example, because you can use also real-life data to sort of put some boundaries around the kind of outcomes that could potentially inform clinical trials.
I would only add that during the course of this meeting, I've been surprised at how much of this is already going on. So it's not like it's coming, it's already happening, I think, number one. And number two, I was hopeful from Peter's remarks is if we could be clever enough to design the right kind of clinical trials, then we could use that to model much more broadly so that we could extend our conclusions more widely.
So I think we've certainly heard that there's a lot of action in the area of polygenic risk scores. I'm not sure we heard, though, that there were like specific trials of polygenic risk scores that were being designed and looked at.
People are just using it.
So yeah, people are just using it. So I guess the first question is, I think we've all agreed that there needs to be trials at some level. So then the question becomes, what kind of trial? And do we need to do a trial for every condition and every polygenic risk score, or do we just need to do some trials that validate that in general this approach works? So, want to take that one on?
Sure, I'll start. I think we actually need to do a trial for every polygenic risk score. It's going to be a biomarker specific and disease specific kind of, because you're going to be doing different interventions based on the different disorders and the different polygenic risk. So it's going to have to be an accumulation of evidence that's going to start somewhere and hopefully inform on other trials that go on after that. That would be my opinion.
George?
I think what I would add to it is we have to make the selection based on what actions we're planning. Knowing is no longer enough. And so selecting where to act based on the potential to intervene would really make a big difference. And I remember a question came up earlier about a wonderful application that goes to steady section, and steady section doesn't seem to get it. I think a really important piece is, so what, based on the findings, are we going to be able to intervene on something? I would use that as an important marker to decide where we go next.
So just to add a little bit to all of this, which I agree with, is that you can think about trials as de novo or opportunistic. There are all kinds of health systems, population-based studies for which we have tons of either whole genome or whole exome sequence data. Even health systems like Geisinger are already beginning to return some of the results for the top genes of public health significance, some of the tier ones that I talked about. So within those contexts, rather than creating and developing new genetic risk scores, testing de novo, you can design trials that are embedded in already collected data and see by selectively returning results versus not returning results to both providers, patients, healthcare systems, look at some of the intermediate outcomes and some of the outcomes that George mentioned earlier for selected conditions. And I would select them very carefully, like breast cancer screening, maybe heart disease, polygenic risk, etc., but not, I wouldn't go for schizophrenia, other conditions for which the interventions may or may not be there. Happy to discuss that more.
Alicia, did you want to add anything?
Sure, yeah. So I would say that there are some places where we can gain some knowledge.
Keep my on too? Yes, sir. Okay. So keep it on.
They'll turn it on and off down there.
Okay, I see. Sorry. There are some leaders that are already starting to explore how this might be implemented in sort of a national setting. So we can turn to some of our colleagues who've been kind of leading this. For example, in Finland, where we actually know a fair bit about genetics, it's a less heterogeneous population than here. And we have some idea already about trials that are ongoing there, for example, from the gene risk studies, where we have an idea about how this would be implemented in the clinic alongside other risk factors, for example, in cardiovascular disease. And there, so far, it kind of looks like folks are more likely to take their statins based on having a genetic risk score, which is exactly what you would predict. Having more information about being at high risk and having your doctor emphasize the importance of taking these statins increases adherence in a highly educated, homogeneous population. Now, I think it's not super clear how that would actually play out whether that would play out to the same extent here, but we can learn from examples of predecessors who've gone before in this space, and I think it'll be really, again, as others have mentioned, phenotype dependent. So cardiovascular disease, we already have a lot of clinical risk factors. We already can measure your LDL cholesterol, and we already have your age and your BMI and all of these things. But in other places, there are fewer biomarkers. So in psychiatric disorders, one place where you've mentioned, we focused a bit on the fact that there are no biomarkers really, and having some sort of behavioral intervention for those individuals at high risk, for example, of an earlier onset psychiatric disorder might actually be beneficial, perhaps in a less druggable way, but still beneficial to the patients nonetheless. So I think it's really going to be phenotype dependent.
David?
So while, in my view, it would be terrific if we had a clinical trial for each phenotype, I think that's impractical. And I think, so that's why I'm hoping that, and not being naive about this, but I'm hopeful that by cleverly picking 3 or 4, we can make a base that we can generalize from. That's number one. Number two, one thing I wanted to mention, and I didn't, is certainly in the population that I deal with, it's a very diverse population, and I think we have to build that into making it the physician uptake and every other way that we use it. And the third point is I come back to the economics. I mean, it's hard enough now to get insurance companies to pay for sequencing. If I had to go to an insurance company and say I want to do a genotyping chip because I'm going to use that information to do a polygenic risk score, and trust me, that polygenic risk score is going to save you money, it wouldn't happen.
So I want to push back a little bit and maybe build a little bit on what David said. So it's a little scary to me to think that you guys think we have to do a separate trial for every condition, and maybe even for every version of the polygenic risk score as you move things in and out. So from the patient perspective, if the patients look at the data and think that there's maybe value, why would we have to go through this process of a clinical trial in order to get there?
Can I take a first crack at this? So thinking about polygenic risk scores in the general population, not from a patient perspective, like everyone in this room, has a polygenic risk score for every disease that we may or may not get in the future. So following Zach's principle of do no harm, it behooves us to think about how we, as people, may or may not want to use our genetic risk scores for heart disease, diabetes, different types of cancers. And from a population screening perspective, when the incidentalome raises its ugly head, we need to do that evaluation. So I understand the pushback, but on the other hand, if you do a proof of concept for at least a few of the major diseases, if you have strong analytic validity and clinical validity, the rest will be kind of riding the coattails if you're beginning to show some health benefits of polygenic risk prediction in common diseases like diabetes, heart disease, and some cancers with screening. The rest will join the crowd as more and more data accumulates. But on the other hand, you can still use those other polygenic risk scores to design trials in high-risk groups for potential interventions, whether they're treatment, behavioral, or pharmacological, et cetera.
We probably should go on record and say that we're not advocating for polygenic risk scores for every condition. We also should go on record to say that we're not proposing the concept of my risk factors are more important than yours, and so we should be doing mine. However, the clinicians have a major challenge in the category of patients who don't meet current guidelines for intervention. If you actually had a trial that had hard endpoints that demonstrated that in categories of people with intermediate risk, you actually can either move down or move up in terms of the intensity of intervention, we think we'll be very interested and it could be very helpful.
Anyone else want to comment on that? All right, well then, one of the things that raises and is the cost of all of these trials that we're going to have to do. So where's that money going to come from? Who wants to take that one on?
It's a very important question.
Yeah, really.
As the nation's lead funder for biomedical research, I think NIH still considers itself a very important player in this field. And so we're not shirking away from that responsibility. But clearly, there isn't enough money to go around to do all the tests that we need. And that's why, and by the way, I didn't say anything about CDC, so let me finish. But it is really true that even at NHLBI and at NCI and GRI, we don't have the types of resources that will go around to do every single important study that we want to do. So there's a real need for collaboration here. And if it's diabetes and heart disease and smoking and chronic lung disease, finding a way to be collaborative and then come to us with that approach can help a little bit. We definitely don't have the resources, not even funding trials at the level we did even a decade ago.
So just to add to what George just said, so we spend a lot of money, meaning collectively as a country, to find genes and develop new biomarkers. A few years ago, we looked at the NIH portfolio in genomic funding, and we found that about 98% of the funding goes to discovery and early translation. About 1 to 2% goes into evaluation and implementation science. So as the basic science and early translation matures from bench to bedside, I think some of that investment will have to follow through, because that's the only way we're going to show translation into real health benefits.
Do we have to have a system where each institute commits to doing N number of polygenic risk score trials? How do we get this done?
Maybe I'm naive here, but why can't a company develop a diagnostic test and then get billed for it in a system, a medical care system? And so it would be a company funding the effort of getting the test done and it would be applicable across different medical institutions.
Well, I think again, the problem goes back to this question of, do you need to do a trial for every condition and every polygenic risk score? Then, yeah, somebody could generate a panel to do that. But isn't it obvious that the thing that we should do is sequence everybody at birth, and that's the way we generate the polygenic risk score?
If it's obvious to you, it's not obvious to me.
Anyone else want to comment on that?
I like it.
Rex, could we pick up on one of these last points? Because one of the questions I was going to have is, I mean, what is the role, or what's going on in the private sector? We really haven't, I don't think we've talked about that very much at this meeting.
No.
I believe there's a lot going on. I don't know how much of it is, I don't know the scale, and I don't know if anybody in the room knows much about what's happening. And are there potentially data in that sector that might be able to be utilized in some way to also help defray the costs of generating it? I don't know. I mean, does anybody really know the private sector landscape with polygenic risk scores?
Iftikhar raised his hand. Maybe he does.
Well, I've been thinking about this a lot. So we did the MyGene study, which was a small pilot study of 200 patients of intermediate risk for heart attack, where we randomized them to a clinical risk score versus a genetic risk score, and we found that the LDL was lowered after 6 months. So for the past 2 years, I've been in conversations with NHLBI to extend that to an event trial. So let me relate my experiences. So the number one hurdle is the cost. At this point, it's a $60 million trial. And NHLBI is saying, can you get the cost down before we can consider it? So we're stuck there. The second thing is the unknown validity of this risk score in minorities. So I mean, if you're going to do a trial where you don't know exactly what the polygenic risk score means in African Americans, it's very hard to cross that threshold. And the third thing we've been discussing is, can somebody in the private sector maybe do this? And we've explored this, but unfortunately, I mean, there are companies that might subsidize the test, like 23andMe, but I don't think there's a diagnostic company out there that would kind of say, okay, yeah, this could be a great thing for us if there's a positive trial, and then we'll make a lot of money out of it. Unfortunately, we haven't found anything yet. So I think those are the two issues, the validity in minorities, and then the cost of doing an outcome study. That's where we are held. And so I think we're kind of stuck because we are not able to move those two areas forward.
I think Dan was next.
So the $60 million price tag, Iftikhar, sort of reminded me that, you know, one of the things that we're doing and other people in there with their CTSAs are doing is we're trying to develop this so-called learning healthcare system, and we've managed to do pragmatic clinical trials involving tens of thousands of people using simple EHR-based data capture tools at REDCap, and mainly REDCap at our place, of course, and you know, electronically based randomization and, if necessary, consent mechanisms. So the $60 million price tag feels like a conventional trial to me, sort of. And whether I think we're in an era where we should be able to use newer tools to develop cheaper, better, faster, bigger, more and more inclusive trials. Mainly based in the EHR, and you know, I look around the room and there's a lot of emerged people here, and that's sort of, I think that's a possibility.
So we have been in touch with the TIN, the Trial Innovation Network, and we're exploring
right.
that.
I mean, that's the right.
I think actually, so I've got, I'm kept sharing hands, but I think next up was Mark.
Yeah, I wanted to respond directly to the question about why don't companies do it. And this has actually been pretty well studied in the diagnostic realm, and it really has to do with the margins. So if you're developing a drug where your likely return on that investment is in the billions of dollars, then for that reason and for the reason that the FDA says you got to do it, you know, you do a trial. But in the diagnostic realm, the return on investment for a successful diagnostic is really very modest, even if you can get the reimbursement for it. And so to invest the types of dollars that are being looked at, it doesn't make any sort of economic sense. I wanted to make a comment and then come back to a question that I'm going to pick on George on. So the comment is that, you know, models have been referenced, which I think is really critically important, because I agree with Dave Alley. There's no way we can do all the trials for all the phenotypes, and so if we accept that, then we have to say, well, which one should we do? And I think that's where models can be really useful, because then you can really say, in modeling, which are the ones that are more likely that polygenic risk score is actually going to have some clinical impact as opposed to all the others. And you can also say, what current data do we have to refine the assumptions of the model, and you know, where are the gaps and what are the key elements to be able to collect. So I think, you know, starting with a model to begin to inform how we rationally choose what we're going to study is important. I'm going to, though, ask my question of George on the hard outcomes issue. And I understand why you like hard outcomes. But as I pointed out yesterday, if you're studying FH interventions in kids, the hard outcomes are 50 years later. And with a 4-year R01, that's not going to, and I think with polygenic risk scores, the time horizon of outcomes that we're going to be looking at, for the most part, is going to be much longer than 4 years. And so how does a 4-year granting mechanism for investigator-initiated research jibe with the idea that we need to capture hard outcomes unless we just fund a bunch more framing kits?
Mark, in fact, I think you answered your question, and that's the type of justification that you need that can persuade a study section. The comment about hard outcomes is actually based on very solid data. Maybe about 7 years ago, David Gordon and Mike Lauer looked at all the NHLBI-funded trials, and the vast majority of the trials that used surrogate endpoints were either not published or they were published after 3 years and had really very little impact at all on changing behavior. On the other hand, even though the trials that use hard endpoints tended to be not as expensive, they were published much sooner and they had an impact. So we are going on historical data, and some of the conversations we have in here clearly point to the future I would completely agree with you that it's not in every instance where you would have the hard endpoints early enough to be able to change practice today. And so providing that type of justification would be very, very reasonable.
Any other panelists want to add anything? Dave?
Well, maybe that's also something that should figure into the choice. In other words, if you could pick phenotypes where there might be a short-term, hard, objectively measured outcome that could be incorporated into the trial, that would be a vote in favor of that condition. For example, coronary artery lumen diameters or something like that. I don't know. But
can I add to that? Maybe this is just hopeful youth, but I would think that if you start with the easiest to pick off diseases and the implementation is in any way sort of straightforward, then the additional diseases that might follow should be even more straightforward from there, because you've already, tackled the biggest practical challenge, which is how do we get a genetic score into any clinical space to begin with. So I think the subsequent, you start, say maybe in cardiovascular disease or in cancer, where we've already got a ton of evidence built up, and then subsequent diseases that you might want to apply this to, I think should ride those coattails and be a little bit more straightforward to implement in some sort of clinical sense. But maybe that's just my naivete.
Well, I think Eric Boerwinkel said earlier that it's not clear that any of these are really easy, low-hanging fruit. So I think the key question would be, what's the first one to really tackle to do that?
Another thing in terms of thinking about the problem, we heard the great story about obesity, and there they took advantage of a cohort that had been followed for a long time and then retrospectively sort of applied the polygenic risk score. And that was very compelling because you had that 18 years of follow-up already built into the study.
I would be remiss in not mentioning family history-based trials, because family history has been around. It's it gives you only the upper, the people at high risk. And we at the CDC did one of the few RCTs on the value of family history in clinical practice by randomizing practices and looking at outcomes in the cancer screening space and diabetes and heart disease with mixed results. So any trials that involves genetic risk scores will have to consider family history somehow.
And of course, we capture family history so well in all of our work. So, Howard, I think you were up next. Howard.
I don't remember raising my hand, but I'll take it.
Oh.
I think
well, then forget it. Eric Boerwinkel's next.
I'm going to switch gears a little bit and challenge us maybe to think a little bit crazier. It just seems to me that if there's going to be a trial done, it's just not feasible to do trials by score and by disease. It either has to be a clustered trial where whole healthcare systems are randomized. But I wonder if we should think about identifying very large progressive corporations. Large companies are self-insured, basically. And it just seems like this is an ideal place where, engaging a very large corporation that would test models of healthcare within their walls is an approach. It's a bit, my idea is not well thought out, but it's a bit wacky. But on the other hand, I think that's the kind of thing that would have traction, because it has a little bit of the trial of the scores, it has a little bit of trial of implementation research. It can be done in pediatrics because their employees have babies. It just seems like engaging large companies that have stability. The problem is some of our largest companies are service companies. They don't have long. So we need to have large companies that have long employment histories, and see if those companies could get engaged in such an experiment.
So it's not such a wacky idea. Rob, you want to say anything about what you've been involved with?
Well, we've actually just had an employers meeting where we brought our self-insured employers into the mix and had that discussion. And I think a lot of us recognize that there's a great opportunity to capture that long-term outcome data. I think we have the challenges with it are each of the institutions have their own vested interests in bringing those interests together to coalesce the data to actually get the outcomes that we'd want would be very helpful. So it's a great idea.
So I wonder if the same could be said for health maintenance organizations, healthcare maintenance organizations that would have a vested economic interest in this kind of trial.
So the way this might work, just building on what I said before, and I showed my PLOS Medicine paper from last year, which some of you are co-authors of. You know, if you go through the trouble of doing whole genome or whole exome sequencing on a well-defined population in a research setting, let's say at Geisinger, Kaiser, or health systems or large insurers, then you might want to start stratifying in a learning health system, putting research and practice together, where you begin to return the results of some of the tier 1 genomic applications. After all, there's about 1 to 2% of people with what we call the CDC top 3 diseases. I mean, FH, hereditary breast ovarian cancer, and Lynch syndrome. There is a lot of things you can do in that tier 2 space, mostly in pharmacogenomics, where you accelerate the evaluation of pharmacogenomic-based applications like clopidogrel and others within a learning health systems. And some of the tier 3, including genetic risk scores, if you have enough large sample size, you can explore analytic and clinical validity in a major way through collaborations before you jump in and doing clinical trials. So you can stratify the kind of research from tier 1, tier 2, tier 3, where you upfront you invest in whole genome sequencing, where you keep a very close firewall between research and practice, and then begin to move things into practice as the data on validity and utility keeps maturing. And if enough groups do it, I think and there is collaboration, I think we can have answers to many, many questions around polygenic risk scores. The difference between polygenic risk scores and some of the current genomic applications that are ready for implementations, like Mendelian genetic conditions, that we all have polygenic risk scores. So it affects every single person in the population, whereas a few of us may have some of these rare or quasi-rare so-called genetic conditions. So the impetus for investing upfront in a research/clinical infrastructure that will begin to move things into practice and evaluate them in a learning health system model is a very appealing idea to me.
One idea, like just following up on that, I think that people haven't really been talking about or I haven't heard mentioned is the fact that all of us is generating a lot of genetic data, and it's already reaching a pretty diverse part of the population, so that might be a pretty logical place to try to start to evaluate some of these questions once the data is of sufficient scale to try to evaluate this.
Yeah, they'll have the sequence already in place and the phenotypes as well. Amit, you were next.
One theme that strikes me from this meeting is really just how good is good enough. And so I think, you know, everybody in this room agrees that we're better at interpreting the genome in 2019 than ever before. But I think we also all agree that there's still room for improvement in integrating with monogenic diseases, integrating with other omics and lifestyle, improving across ancestry and so forth. And I think, you know, the reason it's important is that I think there is a time-sensitive window of opportunity to really do some of these trials and generate the evidence that we want. And you know, I think like right now, it's actually not standard of care to disclose polygenic scores, and that lends itself to randomization approaches. And my take is that this field is actually moving incredibly fast. And you know, if you think about like doing, you know, a trial where you for familial hypercholesterolemia or BRCA1, where you had a biobank at Partners or another organization, and you said, I'm going to withhold this information from half of the people, and the other half I'm going to disclose it to. You know, that's not something that most people would be comfortable with from an ethical standpoint in 2019. And I think, you know, I think there's a pretty good chance we could get there with polygenic scores as well, you know, in 5 years. So I do think there's a pretty narrow window to actually just say, you know what, we have scores that work pretty well, and there's an opportunity now to just study how best to disclose it, do proof of concept studies in the next 2 years before it kind of becomes swept away by other commercial entities or other folks that just move the field forward in a very rapid fashion.
Just to clarify, I wasn't talking about withholding BRCA or FH information. These are in the top tiers. So the genetic risk scores, as you said, we have a window of opportunity to do randomization. But for the other monogenic condition, you do genetic counseling, you do implementation science, and you figure out the best way to transmit the information into a learning health system. But for the polygenic risk scores, I think on an ethical ground alone, we are still okay in withholding the information and evaluating the impact of giving the extra information.
That's exactly right. I mean, the BRCA was used as an example just of something that I think there's agreement that we wouldn't think is appropriate to withhold.
And it's not clear we need a polygenic risk score really there either, right? I mean,
well, I think there's room there as well. I mean, we've generated a fair amount of data recently that shows that actually common variant background modifies the penetrance of some of these important Mendelian conditions. And so if you have an FH mutation, but you have a favorable polygenic score, your risk is actually basically average for the population. And if you have a high polygenic score in FH, maybe it's tenfold instead of 3. And there's a fair amount of data similarly for these monogenic mutations. So I think there is opportunity to explore the interplay between these factors.
okay, I think Terry was next.
Yeah, I think going back a bit to this question of needing a clinical trial for each, we are the Genome Institute, and we like to think omically, and so I think we're in a good position to test a number of these, probably more than 3 or 4. Although I like the idea of kind of stratifying along the lines of one that's very polygenic and another one that has a variety of interventions in that, and it would be helpful, I think, for the panel and others to help us understand what are the sort of varieties of PRS scores or conditions that we would want to include in such an omic approach, to be able to then generalize for maybe our sister institutes who might want to take one very specific example and follow that forward.
One way to start is to look at evidence-based guidelines from the US Preventive Service Task Force and other groups that look at screening and treatments. So, for example, there are evidence-based guidelines around colorectal cancer screening, around breast cancer screening, around osteoporosis screening, and so on and so forth. So you start with things that are already in practice. Then you look at their polygenic risk scores, evaluate whether or not the polygenic risk scores may or may not add some value to this. So, for example, osteoporosis was never really discussed here, but people with family history are at increased risk of fractures, and I think they may need screening earlier for osteoporosis and treatment. So, by starting firmly in practice and where the interventions are working and the polygenic risk scores are reliable, then you can start doing some of the modeling that Peter mentioned earlier. Then all of a sudden you have a list of conditions / interventions for which polygenic risk scores could be evaluated for their added value.
Right. So the question then is how many, are there different types of those? Do we need 5 conditions with exactly the same characteristics, equally effective interventions or equally burdensome interventions or equally frequent or distribution of risk? I mean, it seems to me there are parameters aside from the condition itself that have to do with the differences in how the PRS might be applied, and it'd be helpful to sort of identify those.
Dan, was that a follow-on to this, or?
It is. So I'm reminded here of there's this big pharmacogenetics trial in Europe that is doing not one drug and one gene pair, but many, many drugs, many gene pairs at the same time. So if we're going to deploy an array, and maybe everybody in the room sort of has figured this out except me, so I'll just say it explicitly. If you're going to deploy an array for Boeing or for Northwestern Healthcare System or something, you have the opportunity to develop multiple polygenic risk scores for lots and lots of people. So I would not do one trial for coronary disease or one trial for atrial fibrillation. I'd do a trial where I would pick 20 polygenic risk scores, define the endpoints that you want, and then follow them and give them to half. I would not use the word withhold. I would provide them to half and not provide them to the other half, and then follow multiple outcomes at the same time, it seems to me that that's a design that would help if we did it in a large enough denominator with a way to capture outcomes.
Nicholas, I think you were next on the list.
Thank you. One thing that this is reminding me of is that every time we try to take an informatics method, mostly based on some machine learning algorithm, and we can show it performs really well, and we try to take that and use it in practice, the feedback is always that we don't want a black box, we want to understand, we being the physician, apologies to all the physicians, we want to know, we need this information to make these decisions. Do you think you really can get trust where all these algorithms from other places that are pretty much the same, except instead of based on a whole bunch of positions on a genome, they're based on a whole bunch of other data, are going to be accepted?
Panel, what do you think?
Can I just say one thing? Genetics is much easier than any of the other stuff we discussed today. I mean, I know the problem with ethnicity and ancestry, but it's much easier to measure sequences and making an inference than epigenetics or metabolomics and so on. So if we're talking about genetic risk scores, polygenic risk scores, I think they would go first before any of that other stuff. Because with the other stuff, that black box approach worries me a little bit more.
I think there's probably some more general public buy-in into the idea that your genetic risk factors are important for influencing disease, and there's general interest. I mean, many, many millions of consumers in the US have their genetic information that they've purchased through someone like 23andMe or Ancestry. And the fact that they have this, I think, sometimes encourages them to take it to their doctor and ask, what can I do with this? And I think there's not a lot of trust right now because it's unclear what the value of it is. So obviously we need to assess that value in a rigorous way, which is kind of the purpose of trials, I suppose. But I think the reason that genetics has a chance of overcoming some of the obstacles that other informatics methods haven't is because there's general consensus that it's useful information from the broader public.
I think the next in line was over here.
I'm probably making a point that's obvious to everybody, but I haven't heard it said yet. And I think the problem with these very large, with event trials being extremely large in this field, is going to be a generic problem for all of these risk scores. Not only genetic risk scores, but clinical risk scores too. If you think about a conventional trial that tests an intervention typically against a placebo, the contrast between those two groups is 100% to 0. But generally, when we provide information to doctors, it's only going to affect decision-making at the margin. And you'll need trials. If you want to look at events, you'll need trials that may be tenfold larger than typical RCTs to test decision-making advice. If you're only looking at how you influence decisions, obviously, that's much more feasible. But to go to hard endpoints, I think, is going to be very, very hard almost across the board.
Go ahead.
I wouldn't disagree with you, but I wouldn't use the notion of how difficult it would be to not go there. I think that's the reason why Eric's comment isn't all that's so wacky, right? Truly, as we embed these studies in larger and larger trials and have ways of appropriate randomization in either clusters or other categories, we should be able to approach the rigor that we need without necessarily having to use extremely large or wait a very long time. I truly would encourage us not to be building for surrogate endpoints or building for strictly biomarkers. They don't influence practice, and we have to find better and better ways, public-private partnerships, larger and larger practices to advance true health systems research rather than the older notion of the randomized controlled trial.
But we need to find a way to make it so that it's in the interests of the health systems as well. And
yes.
That's a challenge.
Yeah, and maybe I should add, you know, the problems and the challenges that Iftikhar brought up, we are very, very sensitive to those problems, and we no longer look at opportunities to partner with the private sector as something bad. in fact, public-private partnerships are strongly encouraged, and the more you embed these trials in real practices to reduce the per capita cost, the more excited we get.
Not to change subjects, but I have a question to the community here. How about preemptive pharmacogenomics? I think I heard it discussed one time, because a lot of us are taking all kinds of medications, and you can use preemptive pharmacogenomics in a randomized way to evaluate in health systems a bunch of drugs at the same time. Would that work?
Works at St. Jude, I think.
I thought you might want to comment on that.
What I'm commenting on, preemptive genotyping?
Yes.
Yeah.
For pharmacogenomics.
Yeah, we've been using an array. We started off with the DMED array, and now they've upgraded to PharmacoScan. It's commercially available, run by a private laboratory. And we get all of the results. There's hundreds of genes. They're all CLIA compliant. But our approach is to selectively move those CLIA-approved results into the patient's EHR as we create CDS to support at least one drug for every gene. So we've studied more than 5,000 children. We do it on a study because we think we need to get consent for two things. One, the fact that we were withholding results. Two, the fact that we do come up with incidental findings, and we need to get consent up front about whether the parents and child are willing to accept those incidental findings. And so over 8 years, 5,000 patients, we've yet got only 9 genes into the EHR, supporting about 25 drugs, and we do about 1 new gene-drug pair group every 4 to 6 months.
Do you have data on how often that affects clinical care?
Not comprehensive data. So, I mean, and that's one thing when people talk about, well, collect outcome data. Even if the outcome data are only how often are the genetic data used to actually guide prescribing, it's a completely non-trivial question to ask. For example, proving the negative. If somebody has a CYP2D6 high-risk genotype and I have to prove that they were never prescribed another high-risk 2D6 substrate based on that, it's basically impossible to find that in the EHR. If you have a really smart clinician, they don't accidentally order the wrong drug anymore. They look it up themselves preemptively and don't order it. So to generate even that kind of comprehensive how often is it used data, much less how often does it impact outcomes, is ridiculously expensive, and there's not enough money in the world to do it.
Dan?
I find it odd that the 2 centers for preemptive pharmacogenomic testing are in Tennessee. I know they do that at the Mayo Clinic, they say, but I have no idea what they do at the Mayo Clinic. So we've had a program for about 10 years. We've tested 15,000 people. I'm jealous of Mary because the key to all this is finding people in whom the data are likely to be actionable. Mary treats sick kids who get a gazillion drugs, but it's all the same gazillion drugs. And so the likelihood that those pharmacogenomic data will actually be used in practice and bend the outcomes curve is larger than if you take a large internal medicine clinic. That said, Josh Peterson at our group has published some of the sort of initial outcomes data, with the process outcomes, not the hard outcomes. Lori Cavallari at Ignite and Julie Johnson have put together some clopidogrel data. There's an Italian group that has clopidogrel data. There's a large trial being done at Mayo that should report next year. The big European trial on doing it across all drug gene pairs or many drug gene pairs is ongoing and should also report next year or the year after. And Ignite is currently planning preemptive pharmacogenetic testing. So we're going to have outcomes data, because the difficulty, Muin, is that while it seems intuitively totally obvious to you and to me and to Mary that this should work. I mean, we adjust drug doses based on renal dysfunction all the time, because not only does the label say you have to do that, but if you don't do it, you end up with people with drug toxicity. And there's no randomized trials that ever say to do that. You just do it. And so if you have a patient who you know is not going to metabolize drug X, and you have to metabolize drug X in order to achieve a therapeutic effect, why in the world do you need a randomized trial for that? But that's a sort of secondary editorial comment. So the answer is that we have to have those data because the payers don't want to pay for it. Yeah, they certainly don't want to pay for it in a multiplexed way. They'll pay for one drug gene pair for the drug that you're prescribing today, but they won't do it so you can stick it in your record and use it next year. So that's something that we have to struggle with, and I think some of the outcomes data may help with that.
So can I interrupt my co-chair, or at least now that you finished? Quite all right. Let me remind everybody, this meeting is about polygenic risk scores and risk prediction. We actually had a meeting in this very room on pharmacogenetics. So let's try to shift back to risk prediction.
So next I think was Howard. Can't talk about pharmacogenomics.
For real this time? Yeah. No, nor was it going to be. The question for the panel was, do we need a clinical trial of genomic risk prediction? And I would argue that yes, we need a clinical trial. Most of the biomarkers that are in routine practice didn't have any trials. And they were not pushed into practice. They were pulled into practice because of a drug or something, or an intervention of some sort. And that's everything from BRCA all the way through to cholesterol and whatever your favorite is. And so these predictive markers will not be pushed into practice. They'll be pulled into practice by some sort of intervention. But if we don't do a clinical trial, us eggheads will be whining about it forever. And so we need to do one trial, get it out of our system, and then get on with life. And so for psychological reasons, we need a trial so we can shut up about it.
So Howard,
I would say it has to be a well-designed and a positive trial, because if you don't have a positive trial here, it's going to be a major setback for
That's a study.
future.
If it's not well-designed, then it's really just a study. It's not a trial. So I'm talking about a trial.
So what does that one trial look like, Howard?
I think we picked right now we have outcomes for, let's say, Crohn's, where we could look at things like admissions, numbers of scopes, things that have nothing to do with an intervention therapeutically, but have to do with management interventions. We could do those types of studies and ask a simple question prospectively: do patients who have been identified to be in one of 3 tiers of risk and are managed accordingly have different healthcare utilization than a trial where they're not put into the 3 tiers? You know, there's something like that could be done that would not be a therapeutic intervention other than corticosteroids for admission, but would allow us to understand that, yeah, people that are said to be bad do badly, and get that validation. And then we can worry about the trials, because frankly, it won't be until, troponin was nothing until the interventions with ACS. There was no randomized trial of troponin versus no troponin. There was an example where it was used at WashU and then licensed out to companies, and then the drug companies used it as part of their intervention, and boom. Cholesterol, same thing. Cholesterol was a thing until drug companies made it a thing. And so I would argue that, yeah, we need to do trial, but there's so few other biomarkers in practice that we've done trials on. There's a reason for that. It's not just laziness. It's not just money. It's because we can get there otherwise. Even the Oncotype DX example, they were having revenue for a decade before they did the trial that now has shown that it was true. And then now they get even better reimbursement. They paid back their trial. And so the examples just aren't out there for success for a company pushing things into the market. It needs to be pulled.
Adrian, I think you were next.
Thank you. So we know that clinical trials are expensive. We know that often they're not conducted in representative populations. And so I'm wondering, what are the questions we might answer through clinical trials, number one? We've sort of been skirting around those, but we haven't really explicitly stated those questions. And secondly, can we learn anything from observational studies that might help us focus those?
Panel members, what do you say?
Well, that was on my slide. What are the questions? Right. So I think that's a big. I don't think this panel is going to give you all those questions, but I think someone who is contemplating designing a clinical trial needs to spend a lot of time thinking about what are the questions they're going to answer.
Okay, next, I think back here, you were next.
I just wanted to know, if we're talking about a clinical trial, how big is it? Has anybody done a calculation or rough, envelope calculation of how many individuals do you need in the trial to demonstrate effect? So that, I think that will help people who are thinking in their system, or in their company or whatever. Is this something that we can even begin to look at?
Anyone want to answer that one?
Again, I don't know, this is not my field, but I think it depends on what you're doing. The obesity data was done on a relatively small number of samples, I think, 10,000 or 7,000 or something like that. But other things would be require more. And you have to have, I mean, a related question is where are the controls going to come from, it seems to me.
So for a trial, one of the most important variables for estimating sample size is the effect size. And when we've looked across polygenic risk scores and risk prediction, we don't see effect sizes because nobody's done the intervention. So what you described that Emmett did very carefully in 7,000 people was not an intervention, it was an observation. And the question then is, okay, if you tell those moms that their babies are at the upper decile of risk, does that change things when they're age 18? So that's an effect size that we have no idea.
Right.
And it takes up to 18 years to accomplish.
I mean, you could see it at, you might be able to see it at 12 months or at 3 years.
Well, we heard from the pediatricians that they can see it at, like, what was it, 9 months or something? So
the other, I mean, the follow-up question is, what effect size is worth going after?
Do you want to?
In a clinical sense, yeah.
Yeah, I think my question is specifically for George on the panel. This meeting is not about pharmacogenomics. I don't think it's on. This meeting is not about pharmacogenomics, but you might remember that there were.
I think it's off again. Can't touch it.
You might remember there was a lot of enthusiasm about genotype-guided warfarin dosing, and it explained a large proportion of what doses people were on, and the observation evidence was quite compelling. And I believe it was NHLBI who actually funded the COAG trial, and it actually showed no benefit on heart outcomes. And so, and
read the other papers in the same issue of the journal.
Well, I guess
points.
1 out of 3 studies were negative, and people would dwell on that.
I guess my question is more just what lessons can be learned from that experience as we think about evaluating polygenic scores, or how is that influencing your thought process?
I'm getting a little bit worried that we're really getting into details when we haven't aligned on the big picture issues. The issues around effect size and sample size are crucial. I'm also worrying that we forget Alicia's caveats, the potential to exacerbate disparities. These are all bigger issues. I go to my biostatisticians for their advice on the details, and I would strongly suggest we at least focus on the big picture issue of the need for the clinical trial, the need to pay attention to potential partnerships that will lower the cost for these trials, the need to be really real-world practical, finding ways to embed in practices so that when we find answers, they will be easily adopted, and focus more on the implementation challenges, the barriers, and facilitators. So I'm not an expert, and I can't give you the details about the effect size and the sample size issues, but I hope we focus more on the bigger, broader issues, and maybe Alicia can still talk about it. I haven't heard much at all about the potential for worsening outcomes in population groups who haven't been studied so far, and what we would do a little differently. So,
I would, also pose to the group, like, what broad class of clinical tests or what broad class of drugs to treat a disease work 4 to 5-fold better in European ancestry populations than in African American populations? These are massive disparities, and I think we need to really keep that in mind as we try to go forward and think about implementation. We're kind of getting ahead of ourselves a little bit if we're thinking about health disparities in any sense, where we're already at pretty bad health disparities as it stands, and adding a layer on top of that is really ethically and scientifically concerning, I think.
So if there's a 4 to 5-fold difference, is that a place where we should start with using for the first-in trial?
Yeah, I mean, I think it's absolutely critical that in all of our evaluations that we consider a diverse population that spans the breadth of human ancestry and diversity.
No, I'm thinking even targeting that difference where you're seeing a 4-fold difference and really focusing on the underserved population there as the first in.
Yeah, for sure. We see that across the board, so it kind of doesn't matter where you start, I think. Like, that's a general trend across all diseases and traits that we study.
Terry, I had you as next. Are you all caught up? Okay, go ahead.
Seems like a lot of the discussion is about prevention rather than therapeutic outcomes. And so just to take your point, if you take Crohn's disease, where to me the issue is it's a $50,000 treatment with anti-TNF. So we know that early anti-TNF treatment prevents progression to what will ultimately end up in colectomy. But it only prevents it for 10 or 15% of the population. And that percentage is higher prevalence in African Americans than European Americans. So it really is about a disparities issue. But it's a very focused situation where polygenic risk scores for the response to therapy, not for the incidence of disease, could be applied. And it's got a very hard outcome that's very quick as well. It's either you go into remission or you don't. The potential savings for both the drug company and the health system are enormous. So I think there are a lot of examples like that. But it requires a focus on developing polygenic risk scores for therapeutic response rather than for the onset of disease, which we haven't focused on.
And in that case, do you think the drug company would be willing to pay for the study to do that?
You have to ask Jensen, I guess, about that. But I mean, I talked to the Crohn's and Colitis Foundation and the Helmsley Foundation about it as well, but it's too big. It's too expensive to do a big trial like that. You know, they've not got $60 million to do that one.
Thanks. It's a really interesting discussion. I mean, I guess when I think of polygenic risk, I just view it as another marker of at-risk individuals. And this has been used in clinical trials for years. I mean, if you want to find people who are going to have a heart attack, you take those that are hypercholesteremic, hypertensive, diabetic, obese, and now you just throw on the polygenic risk. And I do think there's also an opportunity, going back to Dan's point of trying to do pragmatic trials. We already have a lot of genotyped individuals. You know, I mentioned that for AFib, we're taking entire populations now and just screening for AFib. That may or may not be cost effective. I mean, pharma's supporting that. I mean, that's a completely industry-sponsored trial. And it's pragmatic because we already have the patients coming to us. That may not be cost effective, but we'll certainly run a trial where we take the tails, where we pull people from our biobank. You take the top 2, 3, 5%, whatever number you want, and you take the two extremes, high risk, low risk, and then you intervene. You have a screening modality where we'll send them a watch or some other form of monitoring, and then it's actionable. So I think you need to identify at-risk individuals, have some intervention, and then have something that's going to be clinically actionable. And we'll do it in a population where if we detect the AFib, it's going to be something where we start them on a new blood thinner. And pharma will pay for that. I mean, that's a new indication. It's a new space for them to be able to sell a drug. These drugs are all on patent, which is why it's motivating. We're not talking about warfarin, so they're still motivated to do it. So I think trying to think of pragmatic trials where you can use the available data in the biobank. I mean, I love the idea of trying to go out and do 20 different diseases at once, but practically, if you don't have a few test cases where you've shown that this can work, it's going to be hard to get those larger studies funded.
So maybe if I can just build on that for a second, then maybe we're not really talking about a clinical trial of polygenic risk score. We're talking about adding that as one component to some other kind of trial?
Well, that's one approach. I mean, it's easy to layer in. It's pretty easy study design. You don't even need huge numbers. I mean, 500 to 1,000 in each arm. Those are very manageable numbers. You can easily make a 2x2 grid where you take high polygenic risk, high clinical risk, the intersection of both, and then you take the opposite extreme, lowest clinical risk, lowest polygenic risk, monitor both arms, and that's your control population of the two extremes.
Any thoughts from the panel? If not, then I think another question that
we would do the trial today if we didn't contaminate our other study that's ongoing. I mean, I think the numbers are such that it's doable. And you won't be able to monitor people forever. You'll have a 6 or 12-month window, and the question is that going to be good enough to detect disease events or so? So I think it'll need to be tailored to specific diseases. Same kind of thing with an intervention for at high-risk individuals with FH or something. It does return of information, modulate behavior in a 6-month time frame or something in that range.
That brings the interesting observation is that for most of us, our polygenic risk scores is not that useful because for many people in the population, we're either one standard deviation above the mean or below the mean. So it's at the extremes of the distribution as Amit presented yesterday. And the difference here is that the extremes of the distribution, the 1 to 2 to 1 to 5%, are still more common than the quote Mendelian conditions. And I think, in spite of all the arguments for and against your findings, there is many more people in the high risk and low risk that you can intervene on or design studies to evaluate potential natural history, progression, and if there are interventions, both screening or treatments, you can evaluate those. But for most of us, probably polygenic risk scores are not that useful.
Dan, it's a little provocative statement in meeting on polygenic risk scores.
So pharmacogenetic variants, but then we started to do multiplexed pharmacogenetics, and we discovered that almost everybody has an actionable pharmacogene variant. So I wonder, Patrick or Amit, because you guys trolled in UK Biobank, how many, if you make 20 polygenic risk scores or 50 polygenic risk scores, how often is an individual actually not average? I agree with Muin that the whole business of personalized medicine applies to people who are outside the average, and most of us are average for most things, but we're not, but all of us are not average for something. And so the question is, how often does that happen? And I guess it depends on how good the polygenic risk scores are for predicting, for defining how outside the average you have to be. But
I mean, I'll just give a short answer, which is at least in our paper with 5 diseases, about 20% of the population was at greater than triple the normal risk for any of the 5 diseases. So in each individual disease was between, let's say, 2 and 8%, but in aggregate for 5 diseases, we got to about 20%. I think very consistent with what you're suggesting for pharmacogenomics.
Again, a multiplexed approach where so you define polygenic risk scores for 100 diseases. But I'm having trouble wrapping my head around what the outcomes would be for each of those.
Yeah, it seems like a conceptually like good idea, but to track 100 diseases at once and figure out what the outcome is really hard. That might even be harder than tracking pharmacogenomic outcomes. I don't know.
Cost of care.
Cost of care.
I mean, right now, if we know someone's at higher risk, we monitor them differently. So that's something that may or may not involve a drug, but just the way we manage them in terms of frequency of seeing them, something as simple as that would be different.
Eric Borwell.
I think I'm building off of what Patrick said, but I'm not entirely sure. I mean, there have been many trials for the common diseases and drugs that we use today. Most of the trials in the last 10 years have some amount of consented stored biologic material. And it seems like, and I'm thinking, I think it was Pradeep and Sake, actually used WASCOPs, and I think one other study, and actually and showed that statins have a greater benefit in terms of clinical outcome, and the clinical outcome, I believe, was MI, first MI, in those with had a high polygenic risk score compared to those who did not. And so I think we could use that as a model and see if we could see what other trials may have power and stored material and consent to allow us to do that for other conditions. And it could be done in a relative cost-effective way. And my other comment to Dan is Mr. Rogers said we're all special.
And he was right. So I think we're starting to get to the end of our time. Maybe I'll give the panelists one last chance to see if they would actually like to amend their starting position or reaffirm that their starting position was absolutely on target. So, please.
So I'm going to stick with my starting position. I think there needs to be a clinical trial of polygenic risk scores. I can amend
a trial
that can look at maybe a small subset of diseases. I could go there. It's not just one disease, but I will also stick with that. I think there really needs to be clear, actionable responses that we're looking for when we do these trials, and it would be behoove us to make sure that that is part of the trial, is that based on stratification of the genetic risk score, we're going to do something differently and we can measure that.
George?
I'll stick with the two comments that I made, but I should take a second to just tell you that when I was first asked to prepare for this, the material I was going to show was from Alicia's work. And I was like, and then it wasn't until I looked at the panelists and realized that she was a panel member. So I wrote to her and said, are you going to talk about this? Because if you are, then I won't. I really think we should pay attention to implementation outcomes, the challenges, because we don't want to fall into the same trap. Framingham did a terrific job and gave us some very powerful tools, and half of the people in this country with high blood pressure are not controlled. And so there are reasons why we don't do as well, but implementation trial designs can help. Building on hybrid designs would be very, very powerful, but I think the most important, we should also pay attention to what Alicia said. So now that I've stolen your thunder.
So my original position was, I think it's going to be phenotype dependent as to whether we need a trial or not, and I would stick with that. And building off of George's point and reiterating a few of the points that have been made so far, I think our trial needs to be diverse. It needs to be reflective of the patient population that we hope to treat. Coming back to the point about all of us as a potential source of data, I think is, this is fruitful and something we should really consider. And echoing again Howard's point, we've been operating with clinical models that we haven't had like a piecewise, I'm evaluating one clinical factor at a time. And so I think we need to keep that in mind when we think about polygenic risk scores. I don't think we need a polygenic risk score clinical trial for every single undertaking that we pursue. We don't need a trial for cardiovascular disease and then a clinical trial for psychiatric disease, and then a clinical trial for breast cancer and each of the different types of cancers. This is sort of a model that I think we need to overcome, and with our first clinical trial of polygenic scores, I think it's going to be really critical that we have clear outcomes and we're thinking about our patient populations who we're actually hoping to treat, and then evaluate after this hopeful success of that trial how to proceed forward from there.
Okay. Just want to channel Zach Kohani a bit. With the polygenic risk scores, epigenetic risk scores, metabolomic risk scores, we have a data-rich environment that we don't know what to do with. There is a simple reason why we call it a tier 3 application, meaning don't do it in practice today. So before we get to the first trial, we need to make sure that we have analytic and clinical validity, and then start doing those trials. Now, I'm hopeful that polygenic risk scores will add value to clinical practice, but they would not be my low-hanging fruits in genomic medicine at this point.
Can I ask Muin to comment on one thing in that comment, which is, what is the relevance of analytic validity if you're doing 100,000 or 1 million SNP chip?
Race, ethnicity, maybe. I mean, are we sure that what we're measuring is appropriate for that population that we want to use that genetic risk score for? And do we get the same results? I mean, I think analytic validity doesn't get studies because we always assume it's correct, but I'm not sure. Clinical validity is even worse.
Tackling your question from a different
So what you're talking about is clinical validity. The analytic validity in this case, as I understand it, or at least as I use it, is does the assay in fact perform? Right. And it's one thing to do an analytic validity assessment of a single gene or a panel or ultimately even to an exome, and I don't disagree with you that we probably have understudied that to some degree. But when we're talking about a SNP panel where we're aggregating across hundreds, if not thousands, of individual SNPs, what is the difference if one is not analytically performing quite as well as the other 999,000? So I guess the point I'm making, to be provocative, is that I don't think analytic validity is relevant in the consideration of polygenic risk score unless we have a panel of polygenic risk scores that's like under 50.
Well, and we all know, I mean, and certainly if they're sequence-based, we all know that those sequence-based tests are very reproducible in terms of what the answer is. Much more so than actually often a single SNP test is.
Well, I would say the SNP chip analytical validity has been, that's a done deal, basically, long ago. I'll stick to my original point. To me, it sounds like an experiment that needs multiple different kinds, 3, 4, 5, I don't know what the right number is, with different architectures and so forth. I do like the emphasis on implementation science, because it's clear that genetic medicine is not going to meet the promise that we all want if people don't understand what we're saying, and if they don't use the information. By people, I mean doctors and patients, if they don't use the information to their benefit.
Right, so I think if then to close it, I would summarize by saying we do need some kind of clinical trial, a clinical trial, maybe of a polygenic risk score. Maybe that should be a test across multiple diseases, multiplexed in some interesting way, or as an add-on to existing trials that are ongoing. I think the idea of engaging, I worry about how we're going to actually pay for this. I think the idea of engaging employers to help us do that in healthcare systems is an interesting one, because they're potentially people that benefit enormously. What do we need to know before planning such a trial? I think that's a big question that we certainly didn't come anywhere near answering. And when we started to venture into the weeds of how big and how many people and what population, I think that's an area that we need to be thinking and talking a lot more about. But I want to thank all of our panelists for their really thoughtful presentations and comments. And to thank all of you for, I think, a pretty lively discussion. So thanks very much.
Great.
And I think now we have lunch.
We have lunch, and we have half an hour for lunch again. If you wanted to make it shorter, we could, but it might be tight because, is this box lunch? Oh, this is box lunch. So actually, we could make it shorter if you want to. So what's the feeling of the group? Go for 20 minutes? Okay. All right. So if we go for 20 minutes, 12:40, we're back in place. Okay. See you soon.